The growth of bacteria can be broken down into 4 progressive stages or phases: lag, log, stationary, and death. We'll now look at each one individually. When bacteria are first introduced to food, they go through an adjustment period, the lag phase. In this phase, their number is stable as they prepare for growth. To control their number and prevent food from becoming unsafe, it is important to prolong the lag phase as long as possible. You can accomplish this by controlling the bacteria's requirements for growth in food, that is time, temperature, moisture, oxygen, and pH. For example, by refrigerating food, you can keep bacteria in the lag phase. If these conditions are not controlled, bacteria can enter the next phase, the log phase, where they will grow remarkably fast. Bacteria reproduce by splitting in two. Those in the process of reproduction are called vegetative microorganisms. As long as conditions are favourable, bacteria can grow and multiply very rapidly, doubling their number as often as every 20 minutes. This is called exponential growth, and it occurs in the log phase. Food will rapidly become unsafe during this phase. Bacteria can continue to grow until nutrients and moisture become scarce or conditions become unfavourable. The time required for bacteria to adapt to a new environment and begin a rapid rate of growth depends on several factors, which will be covered later in this course. Vegetative bacteria, that is those in the log phase, may survive low, even freezing temperatures. High temperatures can kill them. For example, bacteria can be killed by proper cooking. Some types of bacteria, however, have the ability to change into a different form called a spore. The spore's thick wall protects the bacteria against unfavourable conditions such as high or low temperatures, low moisture, and high acidity. While a spore cannot reproduce, it is capable of turning back into a vegetative organism when conditions again become favourable. For example, bacteria in food may form a spore when exposed to freezer temperatures, allowing the bacteria to survive. As the food thaws and conditions improve, the spore can turn back into a vegetative cell and begin to grow in the food. Since spores are difficult to destroy, it is important to cook, cool, and reheat food properly. The graph in this slide shows how different temperatures affect the growth of Salmonella. As the graph shows, at warmer temperatures, 35 degrees Celsius, Salmonella grows more quickly than at colder temperatures of 7 degrees Celsius and 10 degrees Celsius. At even colder temperatures of 6 degrees Celsius, Salmonella does not grow at all, but notice that it doesn't die either, prolonging the lag phase. This is why it's important to refrigerate food properly to keep it safe. Eventually, bacteria population reaches a stationary phase, in which just as many bacteria are growing as are dying. When the number of bacteria dying exceeds the number growing, the population declines. This is called the death phase.